Students, my today's topic is bioinformatics. I am trying to give you a basic knowledge on this topic. It is an information technology dealing with the maintenance and use of data in molecular biology using computers, or in other words, it is the information technology applied to molecular biology. The term bioinformatics originated from two words. Bio means living and informatics means information science. Components of bioinformatics are mainly four components construct the framework of bioinformatics. Number one is genomics, which deals with the study of genomics of organisms. Transcriptomics deals with study of expression of genes through mRNA using DNA microarray. And third one is proteomics, deals with the structure and function of protein. It helps to understand the post-translational modifications, protein interaction, and the three-dimensional structure of protein. And fourth one is metabolomics, which deals with the study of cellular processes and all the components involved in this. Bioinformatics in life sciences, it is very useful, or its contribution of bioinformatics in life sciences is in the genome sequencing. Genome database provides new advances in medicine, biotechnology and genetics, evolutionary studies and diseases like cancer, cystic fibrosis, liver diseases. DNA sequence of different organisms opened new avenues in study of evolution, leading to the study of gene diversity in an ecosystem, analysis of proteins through the sequence folding patterns, secondary structure, domain, and the three-dimensional structure is become easier. As a whole, bioinformatics has influenced life sciences from evolution to nanotechnology. Importance of bioinformatics in our life. It is a multidisciplinary subject which involves biology, computer science, physics, chemistry, mathematics, statistics, and medicine. It deals with the storing, searching, and distribution of biological data. It develops computational tools, databases, and methods for biological research. It provides a graphical interface for biological research like drug designing, protein engineering, phylogenetic analysis, etc. It is highly important in modern molecular biology research, etc. It decreases the duration of drug discovery from 15 years to 3 to 4 years. Bioinformaticians converts the properties of biological system into mathematical models to experiment it in the computer. It has now become impossible to perform a biological research without the role of a computer. Applications of bioinformatics are, it is used in the following fields like molecular medicine, personalized medicine, preventative medicine, gene therapy, drug development, microbial genome application, crop improvement for insect resistant, improved nutritional quality, drought resistant variety, veterinary science, antibiotic resistance, climate change studies, alternative energy sources, bioweapons creation, biotechnology, waste cleanup, evolutionary studies, etc. Biological databases are a computer software containing an organized body of informations on biological data and is designed to update, query, and retrieve biological informations. In other words, a biological database is a collection of biological data that is organized so that its content can easily be accessed, managed, and updated. Some of the examples are, number one is GenBank, number two is PDB, means Protein Data Bank, number three is PIR, Protein Information Resource. So, biology is a data-rich science. Data in database includes biological molecule, nucleic acid, sequence protein, sequence, metabolic pathways, molecular interactions, mutations, polymorphism in sequences or structures or tissue types, genetic mapping, gene expressions, evolutionary tree, etc. Properties of databases are: a database stores data of relevant information. It has facilities for data entry and quality control. It may be formed of primary data or secondary data or other databases. Data files are stored in specific forms. It may be maintained by government or institution, or databases are available to public without restrictions. Classification of biological databases is: it is classified into two types. Generalized database. It has a wide range of related information, is called generalized database. Includes sequence databases and structural databases, for example, DNA molecular databases, RNA sequence databases, protein sequence databases, proteomics databases, structural databases. Number 2 is specialized database. These are created to meet some special needs, are called specialized database. It includes databases of the genomics of various organisms, biochemical pathways, diseases of human being, etc. Functions and types of databases are: data made available to scientists in computer readable form and at one place. Large amount of informations are stored easily and quickly. Data are updated easily now and then. The databases are divided into two categories. Number 1 is sequence databases, containing the amino acid sequence of protein and nucleotide sequence of nucleic acid. For example, BLAST. It is Basic Local Alignment Sequence Tool. And FASTA. It is faster than BLAST. Structural databases contain informations about the structure of protein for RASMAL, Swiss PDB, Viewer, etc. Primary database contains results of experiments, for example, Swiss Prot, GenBank, DDBI, Protein Data Bank. Secondary database is derived from primary database through analysis by SCOP, CATH, ProCite and eMotif. Metabase is a database listing all the biological databases available in the internet. It contains about 1000 biological databases and about 800 web services. Protein databases is a computer software containing an organized body of informations on proteins, which is designed to update, query and retrieve the data on proteins. The protein includes the following components: amino acid sequences, structure of proteins, 3D structure, active sites, translation informations, post-translational changes, location of proteins in the cell. In this database, the information can be updated and an unknown protein can be identified. Protein database divided into two categories. Number 1 is protein sequence databases. It is a biological and sequence database, consists of mainly three components. Number 1, description of protein entry, yani annotation. Number 2, the amino acid sequence. Number 3, links to other related databases. Following are the example of following protein databases. Number 1 is Swiss Prot, UniProt, PIR, XProt, NCBI, protein database, TR, EMBL, etc. Now, protein structure databases, it is containing an organized body of structure of proteins and deals with all the structural informations, also known as a structural database. It illustrates 3D structure of protein with the help of X-ray crystallography. For example, protein structural databases are WWPDB, yani worldwide protein databases, MMDB, yani molecular modeling databases, FSSP, yani families of structurally similar proteins, and CATH यानी classification, architecture, topology, homologous superfamily. Now BLAST. BLAST is local alignment search tool. It is a homology and similarly search tool and biological database contains known amino acid sequence of proteins and nucleotide sequence of DNAs. Its salient features are: find out patches of sequence similarity. Number 2, produces ungapped alignments. Number 3, BLAST is a rapid process. Number 3, it is based on an explicit statistical theory. Number 4, BLAST reports multiple local alignment between the query and the database. Next, program can be run locally or by sending queries to NCBI using email ID blast@ncbi.nlm.nih.gov. BLAST is not guaranteed to detect best alignment between the query and database. Reports from BLAST can be analyzed further with tools such as XNU, SEG, and XBLAST. Uses of BLAST are identification of species, establishing phylogeny, DNA mapping, comparison of DNAs, locating domain organization of proteins, understanding biochemical functions of proteins, studying evolution of species, identification of mutations, identification of specific functions of sequences, knowing amino acid sequences, calculating molecular weight, studying atomic compositions. FASTA by Lipman and Pearson in 1985. It's a technique which is rapider than BLAST. It is a DNA and protein alignment fast homology search software, very much similar to the BLAST but speedy in comparison to BLAST. That's why called as FASTA. It is a biological informatics sequence database similarity check tool. Protein search software is called FAST-P and nucleotide sequences search software is called FAST-N. Uses of FASTA is almost similar like BLAST, but it is very speedy and much accurate. It works on the basis of FASTA algorithm by Fred Tekaya and Geoffrey Barton. Program can be run locally or by sending a query by its email server. This program has high level of sensitivity. I would like to acknowledge Dr. N. Arumugam for this book on bioinformatics. Due to this book, I could prepare this PPT for my 4th SEM students. A basic knowledge of bioinformatics is very nicely written in easy language in this book. Thank you so much, guys.